I'm going to move on and introduce our speakers today. So Karen Yook and Daniela Raciti are the executive editors for microPublication biology, as well as serving as curators for WormBase, based out of the Caltech in Pasadena. WormBase is the authoritative knowledge base for biomedical model organism, for the biomedical model organism C. elegans. And as project managers for microPublication, Daniela and Karen are responsible for the whole project oversight, driving the development of the platform, and onboarding new communities that are interested in microPublication. So for example, they helped onboard TAIR and the Arabidopsis community. As managing editors for microPublication biology, Daniela and Karen vet incoming manuscripts, assign science officers, and manage communications between parties. As curators, they also have established pipelines for data integration into established knowledge bases to ensure data compliance for published research. And today they are going to talk to you about all of these different things. So with that, I'm going to stop sharing and pass the baton on.
Great, thank you, Lenore, and thank you, Ag Biodata, for inviting us to talk. We're very excited to be here. Let me just get my, hope I can do this right. Are you seeing the show or?
It looks good.
Okay, yeah, okay. Great. Okay, so just as you've heard, or maybe you read our abstract, we're here to tell you about microPublication biology, which is our publishing project that we started a few years ago. We did this to relieve the bottleneck in curation for getting published data into community databases. So my understanding is a lot of people here are from databases. So for those of you who aren't, please ask questions. Stop me if I'm being too lingo with the databases and curation. Okay, so wrong screen. So microPublication is our online journal. It's freely accessible at www.micropublication.org. And during this talk, I will tell you more about our journal, who we are, what it is, how we help scholarly communication, and how we help databases. And then hopefully you'll have a takeaway of how you can help us help your communities. Okay, so what is our microPublication journal? This is an online open access journal. We publish standalone single experiment data. They are peer reviewed, and they're also discoverable in Europe PMC, and PubMed. It's a bona fide citation for authors. And also we work with community databases for data vetting and curation, which makes us pretty unique. Okay, so the first article was published in 2016, so we're pretty new. We are nonprofit. However, our funding from NLM stopped last year, and we've started having article page charges actually in April. We're now up to like the third paper. So I'm just gonna get this out there now. It's $250 an article, which is very low. We're still good people, I think. But we do offer waivers as well. So we're not going to turn down any article if you can't pay. And this isn't a plug for money. Okay, so let's see. Yeah, so what is a microPublication? Well, they're peer-reviewed scholarly reports of findings from a single experiment. So you have a bona fide, robust experiment. You've done all the controls. You've done all the, the repetitions. It's a good experiment, but these things, maybe you don't have a place to publish it. So this is a good venue to doing that. So it's typically one single figure and or results table. Text is brief and to the point. You need to, there's some standards of what you should include. And, you just need to do what you do in other articles, make sure your references are there, acknowledgements, funding, and author contributions. Basically anything that's just scientifically sound and passes peer review. Who are we? We are WormBase. We started as WormBase. Paul Sternberg is our leader. He also has a wet lab at Caltech, and he runs WormBase. So as Lenore told you, this is the model organism, authoritative model organism database for Arabidopsis elegans. We also, on our staff, is Tim Shadle. He's an esteemed C. elegans biologist out of Washington. You've met Daniela, and we have just a few more people, one developer basically. So we're very lean and nice. So we do this all. But this also gives you a little hint that as we expand and as we bring in more, meet more communities, we do need help. So as a database, we considered our mandate to increase discoverability of our community's data. That also means that we extract data, we identify data, we annotate data from published literature, and we put it into our databases in context with other like biology, so that our communities can do a deep dive into different pathways and processes, as well as get a broader picture of where things fit. I'm sure that's what all of you do in your other databases. And part of that is also making sure that the data that comes in is in the right format and is recognizable, especially for computational algorithms. So there's a lot of identifying what's being written and translating it into our, with our IDs and making sure the nomenclature is right and so forth. And I think as database curators or people who work at databases, you understand that when you look at a paper, that's not always the case. Authors also, can make typos that can have serious consequences down the line. All of a sudden that gene name is, something else. Also, it's not readily interoperable. It has to be translated into an ID someplace. So there's a lot that a curator does, and it's time consuming. There's a lot of manual effort there. We certainly have striven or strived to automate as much as possible. And I'll show you I'll show you some tools that we're employing to make that easier for us. But there's always going to be some, manual work. And simply, there's a lot of problems with this system. The first problem is that there's gaps in communication. There's a lot of data that we just don't see. I mean, that's kind of a basic problem. Authors or researchers aren't releasing all their data for a number of reasons. These are standalone results, but they show up in papers as data not shown. Well, that should be shown. There are also negative results or reproduced results that don't fit into their publication. Please ignore my cats in the background who are fighting. There's also results that are, they're just not interesting, so they just don't get into the published literature. There's also, results from undergraduate work. We have a lot, how many professors are there out there who run undergraduate coursework? And you bring a nice project to your classes and the undergraduates do some work and there's a solid result there. Well, that gets lost. So how can we help that? Well, we built our system to be able to accept this data. It's modeled after just taking those little pieces of data. So as you can see on our site, we accept negative results. We accept findings previously not shown or replication successful. We have a number of articles in those categories. Here's an example of some articles, what they look like. As I said, they're just single figures. So they kind of have multiple panels. They're just pretty short. Okay, so another problem that I alluded to is that, you know, curation is a manual process, and it's very time consuming. But, you know, as you all know, there's just huge unstoppable growth in research, and the data just is going to get bigger and bigger. But as the data grows, you know, we all know that database funding doesn't. So there's how are we going to handle this? We're already maxed out in what we can handle. So a lot of data isn't going to get into databases. It's good that they're published, but, you know, we know that the use of databases is really putting it into a better context, easier for people to handle, and really validating a lot of the work that comes out and making sure that, again, like nomenclature rules are adhered to or community standards are adhered to, so that these data can be compared with other things and fit into other narratives. And we all know that, you know, curation has to continue because there's always new techniques. That means more types of data. So we always have to pivot to be able to accommodate new stuff. So a lot of stuff is just getting left out. So how can we fix this? Well, our idea is that authors should be curating their own data. So how are we going to do that? Well, our system is getting built to allow that. So what we're doing, let me just show you our current workflow. We are not quite at the point where we're getting the authors in on curating their own data, but hopefully you will see where we're going to go, where we're going, and I will show you how far we've gotten. So our workflow right now is that an author submits their data in the form of an article. Micropublication editors, managing editors like me and Daniela and Leonore look at this article, and we can tell right away if it's, you know, just spam or if it's a bona fide article. We enlist a member of the community that we call a science officer. So the science officer is a well-known person from each field. For us, you know, we're C. elegans. We deal with a lot of C. elegans articles. And so we have science officers from the C. elegans community. Leonore, you work very closely with people from the TAIR community. They are really the people who look at the article and says, you know, yes, this is a bona fide article, it's good science, let's send it for review, and they help us find a reviewer. So the science officer also is somebody well-known in the community who knows, you know, has broad knowledge of other, of their field and of their community members. From there, the reviewer reviews the article. Along this way, anyone can kind of say, okay, well, this isn't really a good article and we can reject it. What we do, which is very different than what other journals do, is we include a database curator in this pathway, in this workflow. So this allows us to get validation from the actual database that their community trusts and that their community uses. So again, going back, if you look at your curation experience, you know how many errors there are in articles and you have to chase it down and figure out how to deal with it. Well, because we have the curator within our workflow, we can get feedback right away so that the curator knows, or so that the author can be told, you have a typo, you can't use that gene name, you have to switch this, you know, you don't have the right controls, we need more information for your article to really fit into our database. So this also allows a database to know that there's new incoming data. You know, maybe the author is reporting a new gene model. The database knows what it needs in order to take that information in. If everything passes and we go through peer review, and you know, everywhere along the line, as managing editors, we deal with all the communication back and forth between all the parties. If everything goes well, you know, author is happy with the reviews and revises the article if needed, then we publish it. It seems pretty easy. But let me go over a little bit more about what these roles are. So again, the managing editor, for us, it's usually a mod or database curator. So somebody, at your database, we would be hoping would help us with that role of managing editor. And again, they go over the initial submission. They make sure that all the relevant pieces are there. They know who to go to for the nomenclature and validation steps. And, they just kind of make sure everything looks like a publication. They communicate, they deal with all the communication between all the parties, like I said, and they just make sure the article moves forward in the publishing cycle. Again, the science officer is a well-known person from the community. The database really has a role in picking who this person is, especially since this person needs to be able to work well with your database. You need to have good camaraderie. They do, the scientific kind of monitoring of the article. And they have the final say in if the article is ready to go or not. And a critical part of this, of course, is a database curator, not just as managing editor, but you can have a different a different curator from your database. And that person is the one who makes sure that everything meets the community standards. And they know what the database needs, what models are needed. And they're the ones who can really guide the author on what needs to be done. So how are we implementing this? Oops, sorry. So I'm going to give you a little bit of a preview of what our tools look like, and hopefully this will excite you into wanting to learn more about our system. This is a view of our editor dashboard. It's got a lot of things going on in it. When the author submits an article, we see the article on one side, and we have all these tools that we can use to go through the article in our own editor panel. One thing one of the first things we need to do when we get an article, even though this is already kind of done, is to assemble the team. So we can assign people to be main editor, to be, a backup editor, the science officer, and the curator. So all these people get alerted to this article is here, and all the information, these assignments and all the chat and all the little steps along the way get communicated to everybody on the team through email, and the email has a link back here. So you never have to really just check all the time. You just, you get a little alert and you are told to, go to the dashboard, or you're sent a link that will log you into the dashboard. So we've made this as streamlined as we possibly can. As curators, we know people's time are very important, and we aim for as much efficiency as possible. But okay, so let me get into something that looks really detailed, but it's incredibly exciting. And I'm just going to give you, this is just kind of a taste of where we're going and how we're doing things. So I had told you before that we want the authors to curate. We can't just lay this on them right now. We're testing this out with having curators do this, but we are going to open it up to authors soon. So one tool we have is an entity extraction and identifier. So I showed you that when an article comes in, it has, our view of the article, as well as the author's view. Our view of the article has two columns. One is the article itself, and the other is this tool panel. One of the tools that we have is called a linker. And when we click on the linker, we choose a keyword set, and that triggers identifying identifying objects in the article. So this is just a view of what happens after the linker has run. you see this article and you see things highlighted. In this column, you get a list of all the entities that were identified. It tells you what type of an identity of entity. It gives you the count of how often that entity is found. And it tells you the source, the ID of it from the database. If you look at the article, you can find these things highlight in a different font. We also have automatic highlighting of, of italicized words. So that kind of gets your eye looking and gives you kind of focus points or landmarks. So just by doing this alone, it's very easy to see if there might be a typo. You know your community's words and entities, and you know when things don't look right, because just this alone like puts your eye at being able to scan and know there's something wrong. You also, this also can easily be moved into an author's realm. So you can imagine that an author is writing and they have access to this linker. And if they run the linker, they'll be able to tell that they have a typo in there. They'll be able to tell if something doesn't exist in the database yet, because there won't be an ID for it. So this, I think, is going to be incredibly powerful for authors. But for now, it's very powerful for us, because the next step from this, now that we have all the entities extracted and we can delete things that are wrong, so we can correct things, we can also add things, and not showing you that part. But once we have a list of entities that we've validated from this tool, we actually have it populate, a little curation form that allows us to make these assertions based on entities that were extracted from the article. So right now, this is something that we're going to have curators do. So, so we have to play around with what entities, a curator wants to find, and how the curator wants to build assertions from those different entities. What is it they need, like how they want this to be modeled. And we're working with all of our different, we're going to be working with all of our different groups to do this. Once that's done, we're going to try to move this into the author's realm, so that when an author submits, they also will be able to curate. Okay, so with that, let me just go back and tell you where we're at in terms of our publishing. So we've started in 2016. Right now we have over 469 micropublications. Over 300 of those are from C. elegans. We don't accept every single paper. They really are peer reviewed, and some of them really are rejected. Some of them, we don't even get past the editors, because they're just, it's obvious they tried, but it just wasn't, it couldn't pass the first bar. And then authors have also, they're able to control if they want their articles published. We now also have 60 in the works. 32, around 30 of them are actually from a genome education project. So we work with different undergraduate groups, trying to see how we can help them get their data out. The average turnaround time is possibly about a month. We just went through a little hiatus as we built and released our new website. But things can move incredibly fast. We've had one publication that came in and went through review, went through revisions, actually a few revisions, and it was published within 8 hours. So it's possible. Everybody's in the right time at the right place. It can happen. Getting reviewers is, just like with every other journal, getting reviewers is can be really long step. Leonore has some experience with that. Okay, so we have expanded. Certainly Daniela and I can't have done over 400 articles by ourselves. So in the worm field, we've brought in more people to help out as science officers and as managing editors. We don't just do worms anymore. We have fly papers. We have people from Flybase. We have zebrafish papers. So Monty has stepped up from ZFin. We also have Saccharomyces cerevisiae papers, and we have people from our colleagues at the SGD. We also have people from Frog, Dictybase, and PomBase. And those papers, we don't have a lot, but those are coming in. And this will look familiar. Leonore graciously helps us as a managing editor from TAIR. We have a couple science officers, and these other people you might be familiar with, Lucas Mueller from Soul Genomics, and Lisa and Carson from Maize GDB. Hopefully you will, this has enticed you to come talk to us. Maybe you would like us to help your communities. And if that's the case, we want to talk to you. We need your help, from the database side to make sure that we have everything in place. Here's our contact information. And I also have to acknowledge our publishers, which is the Caltech Library. Okay, with that, thank you for listening. I hope that wasn't too boring. I
thought that it was great. So I want to open the floor up to questions. If you want to put questions in the chat or unmute yourself, that's fine. Is there anybody who has an immediate burning question? You can raise your hand, I can call on you. So I guess I'll start with the question. So so for people who are thinking about joining in, from a particular database, like how does one go about getting your particular bioentity set included in the selection process? So we know that gene names are not necessarily even unique to different species. So so in order to get that list of, let's say, like maize genes or soy genes, how does one go about like providing you with those really important identifiers?
Well, we need to know, we need basically a flat file or where we can grab these objects from. That would be the easiest. Our system requires the ID, a public name, so the name that the author is going to use. Having other information like synonyms and official names will help as well. We need to know what one part of this is also linking that object back to your database. We don't have the hyperlinking in our articles yet, but that is what's going to happen. So we need to know how to link back to your database using those IDs. So that's just a URL constructor. We need to know what rules you have for your genes or protein or those names. In C. elegans, we have very strict formatting rules, right? C. elegans genes are all lowercase. In a text, in an article, there also should be, it's lowercase 3 to 4 letters, italicized, and 3 to 4 letters, a dash and a number. That's a typical, regular expression for those genes. So we can run a predictor on that article to find things that fit that. And that gives us a list of genes that you can easily say, yes, that's real, So we need those rules too. So if you have those, it could be just, for every protein, you use the same gene name, same gene format, you just add a P, something like that. That will help us.
So there's a question from Laurel Cooper asking about, I guess, how you're using, or are you using ontologies in the extraction?
Yeah, and Daniela, step in anytime you want. So we do. So for the, we have two steps to this linking. One is just, easy schmeezy, like string match of known objects. And that's not, we're not going to use gene ontologies or ontologies too much there. That's just making sure that typos, don't exist. And then the curation part, we have a different set of terms we run linking through. It's more comprehensive. It has more like anatomy terms and phenotype, possible terms and life stages and things that you want to use to build your assertion, your annotation. So those do use ontology terms, and we have those. So we have those lists, and we have, ways of capturing, it doesn't exist in ontology, then you need to be able to put it in and say, hey, can I get this term, or at least a placeholder so to go back and do that.
Thanks.
Yeah, okay, sorry, I just wanted to add that in the, so the management of the lexica is really cool because, for example, life stages for C. elegans, our ontology term is L1 larva CE, and no author is ever going to use that term. So we can, if they're just using L1, then we're putting in as a synonym L1, so that for future articles, these terms will be recognized and will be linked. And it's really easy to do.
Yeah, I'm really excited about this tool. I have to tell you, like when you showed that to me, I was like, yes, this is great. So next question for Monica, and then Sook. So Monica.
Hey, thanks. Yeah, and I think I might have 2 or 3 questions actually, so if I may. So the first question is, so thanks for the presentation. This is really fun to see. I work for an insect genomics database, so we're just, all these new genomes that are sort of nascent, and we are really grateful for the model organism community to have figured out some of these standards and these problems for us first, and so we might be able to hop on the bandwagon where things are already developed for us. So we shall see about that. My questions are, first, this is great that you are doing this. Within AgBio data, we're also thinking about how to establish relations with other publishers and journals. Do you think that this linker software could also be, and your model, like your model for editorial process, could be adopted by other journals as well? That's my first question.
I hope so. I know Genetics and G3 already uses our linking, so we built a whole linking system for those articles that works on, Genetics and G3 XML. That's kind of the limiting step. It's what that input file looks like. We've taken that tool and adapted it to work in our system, in our platform. In fact, the same developer helped on both projects. So the linking itself is really easy. It's just string matching, and it's just, there's no special, there's nothing special, as long as the hard steps are the file that you're using, and then making sure the links are accepted into a publisher's system. So publishers have such static, solid systems that can't pivot. It's really hard for them to want to do that because it takes time. Because, if I'm sure you've, Europe PMC, I think, has kind of a linker system. If you look at their articles, I forget, but you can go and you can mark it up, and you can mark it, using a whole bunch of different vocabularies. But you need a person to make sure that those links are valid, are actually true, because of, the ambiguity across species, across, who Go terms are not human readable much of the time, but they'll link something. And then genes, if you don't know the organism, it's just going to be wrong. So our system with Genetics and G3 is they send me an article, they say it's a worm article, and I link it with a worm entity list. genetics and G3 are just phenomenal, but, you know, it doesn't, I don't know what, you know, they only cover so many organisms. It would be, but it has been really useful because I have corrected things for authors before they've published, and it, you know, it really works well. Hopefully, you know, but if we have an authoring tool that can do that, then maybe that will, you know, that will help offset, you know, the publisher's unwillingness to change their systems to put this in. But, you know, you can always, if we can band together and, you know, protest, maybe the journals will listen to us. Wouldn't that be great?
I think that's one of the sort of highlights, right? Is that, you know, this is a project that was born out of the, you know, the needs experienced by curators. I think, you know, those of us working on databases can all sort of relate to that, like, what gene is this? You know, or as I made the comment about, you know, even the string matching bioentity recognition for things like PubMed they're not species specific. And so you get a lot of like, that's just the wrong gene things. There's a question for Sook from Sook. I'm curious.
Follow other questions, I think, or.
I'm sorry, do you ask your follow-up?
Sorry, I'm hogging the airwaves here too. My next question was, I noticed that you had a new APC charge due to the funding from NLM going away. How do you think that will affect your ability to attract manual curation from authors?
That's a good question. I think they're going to like it. I think they're going to feel like they're getting their money's worth. Because whoever looks at that stuff, I mean, we know that people don't, even reviewers, you know, they don't catch these things, and they're the experts, right? It's like, this is your field, and you're willing to, like, oh yeah, I know what they mean by, you know, when they swap the letters around. It's like, yeah, they mean something. They don't, you know, I don't know. They don't see it. So,
yeah, we find errors in alleles and typos in alleles genes all the time, only when we are curating, because we're using controlled vocabularies, because they don't appear in the lists. So the reviewer really like just takes a look at the article from a scientific perspective. They don't go double check if, you know, that identifier really had a 0 in there. But then when you're trying to put it in the database, it doesn't work. And then, as we all know, curators, like post-publication, we have to track back the author. They don't want to issue corrigendum. And it's like, forget about it. But then the error is perpetrated in the future. And I agree with Karen. I think the authors are going to benefit a lot. It's like a biological copy editing for the price of, I was about to say for free, but yeah, it's for the price of the micropublication.
I mean, authors are embarrassed when they have those mistakes are pointed out to them. I know that, and you know, they don't want that. They don't want, and journals should be, you know, embarrassed too that, you know, they're missing some kind of big things there.
So Sook had a question about if what you've observed that authors might be more reluctant to publish a micropublication since they wouldn't be able to include that experiment in a larger publication later on.
But actually, it's so much more beneficial to them because, one, their data gets out. It's citable now. They have to reference, you know, anyone who uses that data has to reference them. So they don't lose out on, you know, provenance. They don't lose out on being able to talk about that data someplace else. They can't publish that data again, but they can reference it. It's just like any other normal citation. So I don't see a downside to getting it out.
Yeah, I mean, I would say that, you know, most of the, you know, things that we've seen are, you know, like phenotypes maybe that people have sort of like seen in, anecdotal, not anecdotally, but incidental to maybe some other larger project.
Yeah.
And again, there's also like that cures component. So if you have a undergraduate research experience, then, this enables that publication. Rex, do you have a question?
I had a comment about Suk's question. The point is that if you put in a micropublication and then, which is to use a pejorative from long ago, it's an LPU. And so I think what Suk was saying is, well, I've got one LPU over here and I got another LPU over there. If I put them together, I might have one PU. And that is that seems to be what Suk's getting at.
What's an LPU?
Least publishable unit.
Ah, well, it's no longer a least publishable unit. It's a PU, and you can go poo all you want after that.
I think it also depends on, if this is a key experiment that will build your science paper later on, you're not going to micropublish it, right? You're going to keep it for your bigger story. If this is something that it doesn't hurt that big paper with a big impact factor, because unfortunately, impact factor is still a metric for success of researchers, then you just put it out there and you share it with the community. So it really depends on the piece of data and how is important for your lab's work.
There's a question. Oh, sorry.
No, go on.
So there's a question from Jonathan Sears about, do you validate the references or DOIs through Crossref?
Did you win it? So our submission form takes PMIDs and DOIs. Right now, when you input a PMID, so you're supposed to enter the PMID, our system looks that up and auto-populates the citation. So maybe that's not what you're asking. Am I addressing
No,
it?
that's exactly what I'm asking. Yeah, thank you. Just curious, because so many times you click on a DOI and it's not, in some major publishers, and it doesn't resolve. So that's great.
Yeah, I don't know if our DOIs resolve, but the PMIDs do, and PMCIDs also resolve.
Thank you.
So I was also going to raise something that maybe, for some of the people here in this audience too, about, I think that at least for Arabidopsis, and maybe this is, why Maize is interested in this as well, is that there is sort of this historical community around this, I wouldn't say necessarily micropublication, but like for Arabidopsis, there was Weeds World. So, there was this sort of small publishable unit kind of enterprise. And, for Maize, I know there was the Maize newsletter as well. Stephen Ficklin, you have a question.
Oh, yeah, sorry, I didn't mean to interrupt your thought. Were you done?
It's okay.
I was just hoping to wait my turn. Yeah, so I don't have a database that I manage myself, but I do work with a lot of folks here who also developed the triple framework, which is used by a lot of the databases in AgBiodata. And so this is kind of a different type of a question, but one of the problems that we've had recently is, I think, probably similar to what you're trying to address with the micropublishing, is that a lot of times we'll have developers who will create infrastructure for the databases, and that can be shared by multiple databases and used. But then they don't know where to publish it, or they can't create a full publication, or if they shrink it down, it becomes too cost prohibitive just to publish that small thing. And so I'm wondering if there's been any discussion or interest in maybe going kind of the opposite direction too, where having databases have the ability to publish just small micropublications describing functionality or features, and maybe describing formats that the data needs to come in terms of, in order to be able to make that take your data and make it available within that functionality.
Yeah, we do. We already have done that. Because, we also develop software. So I can share my screen again and show you. So, so we have article types, we have software, and we have data updates. Oh, software. So these are things that here's a tool that was built by developers by people at WormBase. This is actually designed by curators wanting to look, to expand on Go enrichment or phenotype enrichment, basically ontology enrichment. So it can be written up. We also would like to do regular database tool updates, as we get new versions or we update things, or especially to handle new types of data, like new technology, how do we analyze that? What tools are we making? This is the perfect venue for that. We don't need, we will archive your GitHub repo. So we're set up for taking software, and we just need to know version because we really want these things archived correctly. Does that?
Yeah, that's perfect. That's great to know. I didn't realize you had that. So I will I'll advertise that amongst our group.
Yeah, I think it's really sad how much, databases do that just aren't getting acknowledged, right? People don't understand. And the tools you create for your community, this is the best way of showing them how to use it in a way, just alerting them to it, because we don't, it's really difficult. And we can take movies if you want to also do tutorials, but yeah.
Okay, thank you.
Thank you so much. We have one more question from Sook, and I think that'll be the last question. And then I'm going to encourage folks to reach out directly to Karen and Daniela if they want to follow up anymore. And I'm also happy to answer any people's questions about what it's like working with them. It's great. And, other things related to being a database curator on this project. So Sook?
Yes, I just have a related question to Stevens, and I'm wondering, so to publish our database products and functionalities, it needs to have, so it needs to be something about WormBase or whatever, whoever does this micropublication, right? Or do you publish any other database product?
We just need to know if it's something that's very specific for the Maize genome, we would want to have Maize involved and the Maize database involved to help us find people to review the article. If it's just a general tool, I think it's, we do end up accepting things that falls out of our realm of what we're currently set up with. We only have so many collaborators right now, and we want to expand. So every article that comes in, we try to make sure we have the support of a database, but there are, of course, articles that come in, and then we don't. So in that case, our PI is quite liberal with saying, yeah, we'll take it. And then hopefully if we get enough, then the database, the community database understands that, hey, maybe we should help them. But I don't know. We really want to go from the database community support to then getting articles, but that, it's the chicken and egg kind of thing. Why would you, in terms of your question, it sounds like you have some software that you've developed for, I don't know which database.
Our community, Raciti,
Oh, yeah.
Then,
Yeah, the only thing I can suggest is just send it to us. The worst that can happen is saying, no, we can't handle it. We can't handle it right now. And maybe you can help us get a community together, who can handle it. And think about who benefits from that software, who's going to be using it. Is it really just directed at the Raciti community, or can other people use it? And if so, then that expands who we can ask to help us.
Okay. Thank you.
We're not limited to worms. In fact, you know, we kind of got worms covered. We need to expand to other groups, because I think you guys know, I mean, it's so difficult for your communities to find places to publish. You know, there's just, so we need to get those people, their data is being left behind.
So I wanted to thank Karen and Daniela and all of you for attending. And again, you can reach out to them directly. They've set their email in the chat.